I would like to welcome Krista Williams, ABMI's bryologist, to tell us about the wonderful world of bryophytes and why we monitor them across Alberta.
Hi, everybody. Thank you, Sydney, for the intro. And thank you all so much for joining me here today. I am so excited to talk about my work at the Alberta Biodiversity Monitoring Institute and why bryophytes, which are a group of plants, they consist of mosses and liverworts, are worth monitoring. So before we begin, I'd like to try something a little bit different. I'd like you to ask yourself, how often do you notice moss? When was the last time you took a close look at moss? Aside from the bryologists and lichenologists in the audience, when was the last time you put your nose close enough to the ground that you could smell the earth? Have you ever noticed that moss is around us in almost every place that we go? So my goal here today is that the next time you are out for a walk, that you can't just pass by moss without thinking about what these tiny plants do for us, what they do for our ecosystems, and what they can tell us about our biodiversity. I can't wait to explain why I'm so fascinated with this group of plants, but also more importantly, why you should all care about them too. But before we dive deep into the bryophyte world, we sort of need to zoom out a little bit first. So here we are. We're living in this amazing environment. It's full of life. These ecosystems that we are a part of, they are complex. They are diverse, but they're also constantly evolving and changing. A landscape that is full of animals, plants, insects, and microorganisms, lots that we can't see and many that we don't even know about yet. This is our biodiversity, quite simply defined as the variety of life. And we, humans, are a part of this biodiversity. We are connecting with it, we're interacting with it, and of course, we benefit greatly from it. It provides us with our clean drinking water, raw materials for food, shelter, clothing, and medicine. And aside from the intrinsic value of biodiversity, we should all care about it because without it, we don't have these ecosystems that are so critical to our health and well-being. But it's impossible to ignore the fact that our ecosystems are changing. Our climate is changing, our biodiversity is changing. And we need to track these changes so Albertans can make responsible decisions. So at ABMI, we like to say it's our nature to know. But what do we need to know to make these better decisions? Well, it starts with gathering information, and not just any kind of information, but the right kind of information. Better information equals better decisions. So as I said, ABMI gathers information on biodiversity, but we can't measure everything, this variety of life. So we must carefully select bioindicators. This is a species or a group of species that can act like a thermometer and provide insight into what changes are happening and the health of our ecosystems. Bryophytes are one of these indicators. But for many of us, bryophytes are just part of the backdrop. They're small, they're inconspicuous, and easily overlooked. In a forest like this, they're almost like a wallpaper or a carpet. And we go in these hikes in these spectacular ecosystems, or maybe it's views like this, and we're not inclined to stick our nose to the ground and look at these bryophytes. But I am hoping to convince you today that we can make better decisions about our ecosystems with help from our little bryophyte friends. We can make good decisions when we use good information from solid indicators. So the scientific evidence is there and has been for some time on the use of bryophytes as indicators of ecosystem health. Hundreds of publications outlining how bryophytes can be used as indicators of changes in habitat, climate, forest integrity, and pollution. But it's not just the scientific communities, Indigenous communities, naturalists, everyday Albertans, they do recognize the value that these unique plants bring to our ecosystems. Peatlands in particular have been in the news quite a bit lately because of their link to climate change, and I'll talk a little bit more about this later. But before I get into the specifics of the bryophyte component, let's just quickly talk about what makes a good indicator in general. So there are many different criteria that one can use, but here I present some of the more commonly agreed upon ones. So they must be broadly distributed and speciose. They need to be efficient to monitor with intrataxon diversity. Species are expected to respond to a broad range of stressors. They need to be taxonomically tractable. So the taxonomic expertise either exists or has the potential to be developed. And finally, together, the suite of taxa being monitored should represent both temporal and spatial variability. So each taxon that ABMI monitors is hypothesized to provide a different view of a changing Alberta. So what does ABMI monitor? Birds, mammals, plants, bryophytes, lichens, and soil mites on the terrestrial side of things, but also aquatic invertebrates on the wetland side of things. So what is it about bryophytes particularly? Why are bryophytes a good indicator for biodiversity? Well, firstly, they are a vital piece of this biodiversity. Our ecosystems would not be the same without them. And secondly, they provide indispensable information about our ecological integrity. So I'm going to actually start with probably a little 1-minute bryophyte biology lesson and why it matters in the context of biomonitoring. So here's an image of a typical leaf in cross-section. So these higher plants are vascular plants. They have a thick waxy cuticle on the surface of their leaves and water conducting tissues, xylem and phloem, throughout the plant. So when it rains, you can easily picture how a raindrop rolls off the surface of a leaf. And this is because of that waxy surface. It prevents water absorption. And instead, water is brought up from the ground by the roots and distributed throughout the plant by the conducting tissues. Bryophyte leaves look very different. So they, first of all, are so thin, they're nearly transparent. They do not have the waxy cuticle and water conducting tissues typical of higher plants. So when compared side by side, now again in cross section, you can see moss leaves, they are typically 1 cell thick. So when it rains, water is absorbed directly from the surface of the leaf. And not just rain, but ambient humidity and moisture in the air as well. And they absorb nutrients this way. But because of this, any pollutants that maybe come in or blowing in on the wind, they also stick to the surface of the moss leaf. This makes them fantastic indicators of pollution. I'll talk a little bit more about that later. So because they absorb the moisture directly from the surface of the leaves, this makes them highly susceptible to changes in moisture and light conditions. So for example, bryophyte community composition changes significantly in Alberta's boreal forests as a result of seismic lines and well pads, not just on the line or the well pad itself, but several meters into the surrounding forest, where soil temperatures are significantly higher, as is light. And this is reflected in an overall lower species diversity. However, their inability to regulate and store water has not hindered their success, and they are not restricted to wet places. So in fact, they are incredibly successful. This is thanks to poikilohydry, which is a fancy way of saying that they can dry out and lay dormant until more favorable conditions return. Here's a little YouTube video of star moss in Trichia borealis. It's not playing. But what happens is basically as the water hits the moss, it really starts to open up and it just completely unfolds. And I never get bored of watching this under the microscope. I was hoping to share it with you. There it goes. So this is almost in real time. And it's just getting itself ready for photosynthesis. So this feature, poikilohydry, is another biological trait that makes mosses a great indicator. And that's because they can grow in some of these harshest conditions where most plants simply could not survive or establish. And mosses don't just establish in these environments, they thrive in these environments. It is important to note, though, that species that tend to grow sort of in those moist environments are usually not the same species that grow in these rocky and dry environments. So this means that bryophytes can provide information where other flora and fauna are not available. And As I've stated, they occur on nearly every substrate in every natural region of the province. And we have such a diversity of ecosystems here. The Rocky Mountains, foothills, Canadian Shield, boreal forests, parklands, and prairie regions. But they also occur in our cities and our roadside ditches as well. So bryophytes are really resilient and tough, but they have this intimate relationship with water. How can you do both? Well, that's why it's just important to remember that different groups and species grow in different habitat types. So, for example, the knight's plume on the right there is a forest moss. It typically grows on soil and humus, but also really likes rotten logs, and it can be considered a generalist, a habitat generalist. While others require very specific conditions. For example, the rare Porcel's bryum grows in the mountains, and it only grows on protected calcareous cliffs that are shaded and receive consistent moisture. So this leads me to the next topic of bryophyte ecology, also hopefully in 1 minute or less here. So when discussing bryophyte ecology, I always love to think of the term the biosphere. Lindo and Gonzalez in 2010 defined the biosphere as the combined complex of living and dead moss tissue and its associated organisms, representing a tightly coupled system of both above and below ground processes. Their motivation to define the biosphere came from the growing recognition that these systems can have a strong influence on the structure and dynamic at the ecosystem level. So most mosses are just a few millimeters, like liverworts are even smaller. A full stem can be like the size of an eyelash. They are adapted for life in the miniature, and they live in what is sometimes called the boundary layer, this small interface between the land and the air. So they live in this small microenvironment, but yet they have entire ecosystem scale influences. So let's explore that a little bit by discussing ecosystem services. So first, they are habitat. They are these microcosms of diversity, providing habitat for invertebrates, algae, bacteria, and fungi. They truly are like a little miniature forest that is full of life. So some of these organisms are only found in bryophytes, whereas others just maybe sort of frequent it once in a while, do a little shopping before heading home. Bryophytes provide food and nesting material for birds and small mammals as well. The small mammals do actually eat the little lipid-rich capsules, the spore capsules on the moss, which is actually really annoying for me because I'll get a sample and all the little capsules are gone, and often we need those for identification. Bryophytes play an integral role in decomposition. Fallen logs in particular become bryophyte rich as part of the decay process and provide valuable substrates for further biodiversity. So as I stated, they provide habitat for many organisms, but none is maybe more important in the decomposition realm than the habitat it provides for fungi. Leaf and stem structures of the moss can provide these little miniature niches for the fungi where the humidity and the pH and the conditions are just right for certain groups to grow. So bryophytes can colonize, as I said, these inhospitable substrates like a hot and dry rock where fungi would otherwise be unable to colonize. But because of this intimate relationship, nutrient deficient sites can be colonized by symbiotic associations of bryophytes and fungi. Another fine example of habitat niches are a group of mosses commonly referred to as the dung mosses. So moss spores usually, for most groups, are released from a spore capsule and they are distributed by the wind, the spores. But this group, they are what's called entomophilous, meaning that their spores are dispersed by insects. No other seedless plants do this. So these sporophytes you see here, they are mimicking the flowers of higher plants with bright colors and odors that actually attract flies. And then they go from dung patch to dung patch, or in this case, decaying corpse to decaying corpse. A major component of biological soil crusts, bryophytes initiate primary plant succession. So for example, cord moss or umbrella liverwort are often some of the first plants to establish after fire and colonies can cover very large burned areas. This is critical in helping prevent soil erosion and increase its moisture retention, but also improves the quality of the soil, paving the way for other plants. So as I said, I can't talk about everything. So these last few points, I'm just going to sort of list. They are major contributors in productivity, biomass accumulation, nitrogen fixation, and nutrient cycling. So this is actually a good time for me to talk about peatlands and the many ecosystem services they provide. I could do an entire presentation on peatlands alone, but I'm only going to be able to touch on a few things here today. So to me, these are one of the most beautiful and valuable ecosystems on Earth. They are valued for their biodiversity, their importance in global geochemical cycles, as well as the paleoenvironmental records they preserve. They are like a carbon warehouse, removing carbon from the atmosphere and storing it deep in the layers of organic soil. Boreal peatland ecosystems cover about 3% of Earth's land surface, but contain about 30% of the world's terrestrial soil carbon. So what are peatlands? Well, they're wetlands that are comprised of accumulated peat, which develops when net primary production is greater than decomposition of plant matter. One of these plants is sphagnum moss. So here I want to present the province-wide ABMI wetland inventory in partnership with Alberta Environment and Parks and Ducks Unlimited. Peatlands are quite predominant in Alberta. Wetlands cover approximately 20% of Alberta's surface area, and 90% of these are peatlands. I encourage you to go to our website, abmi.ca, and check out the full report. So there are several classes of wetlands in Alberta, but I'm just going to talk sort of about two. So the first is bogs. Bogs receive most of their water from rainfall. They are very acidic with a pH of 4 or less. And they are often regarded as these hostile habitats with low diversity. And in some cases, that's true. But the variation in moisture and the structural diversity of bog vegetation can actually make them more diverse than the surrounding habitats. Rusty peat moss is a perfect indicator of a bog. If you find this moss, you know you're in a bog. So on the other end of the scale are fens, which are groundwater fed and are nutrient rich, much less acidic with a pH of 5 or greater. Warnsdorf's moss or the woolly moss are also perfect indicators of fen habitat. Peatlands offer one of the only ecosystem services with an economic value. So Alberta started harvesting peat in the 1960s. Peat is harvested and exported abroad, primarily to the States for horticultural uses. And in 2013, sales values were estimated at about 69 million. Peatlands also have tremendous cultural value. They are part of the traditional territories of many Indigenous peoples across Canada who remain deeply connected to these places for their beauty, food sources, and medicinal values. They can be biodiversity hotspots, supporting many mammals such as moose, birds like the red-winged blackbird, plants like the sundew, or that adorable little mite in the bottom left-hand corner there, which is only found in sphagnum moss. There is a larva picture there in the middle. It's the northern caddisfly, which ABMI has picked up a couple times. They live exclusively in acidic bogs, and they actually wrap themselves in this protective casing made of moss. Always a fun find is the carnivorous pitcher plant, but let's not leave out the mosses. So here is a beautiful photo of Sphagnum warnsdorfi with its pinky purple leaves that are all ranked in a row with its neatly arranged stems. Here's a view of it from the top. I never get bored of seeing this guy, and I see it a lot. But it's not just sphagnum moss. Other mosses as well, like this very cool angled Paleudella moss, or the beautiful reddish brown color of the hook moss. Both of these are typical of rich fens. So what role does moss play in these peatland ecosystems? Well, they are the ecosystem. They don't tolerate it or adapt to it, they create it. They are the foundation or a keystone species of these incredible ecosystems. And peatland experts agree that comprehensive peatland mapping and monitoring are urgently needed. So I mentioned that peatlands are a carbon warehouse. I'll just briefly touch on that. Atmospheric CO2 is fixed by plants via photosynthesis during the growing season, and it's deposited as litter both on and in the soil. It slowly accumulates and the carbon and sequesters it over time. But this process to build up these carbon stores is slow and takes hundreds to thousands of years. And then when peatlands are disturbed, the carbon can be released quickly, making the peatland a carbon source to the atmosphere. Peatlands also play vital roles in regional hydrology, both in terms of water storage and water purification. So I've established, hopefully, the vital roles that they play in our ecosystems, why we should care about them as part of our communities. But let's get back to the basics of what makes a good indicator, this time specifically with regards to bryophytes. So first was broadly distributed. I've covered this already. They occur in every natural region in the province and can grow where other organisms cannot survive. Rare species are an important part of this biodiversity. A very recent study published by Renee Belland and Richard Kainers examined patterns of rare moss diversity and distribution in Alberta and included the use of ABMI data. In our province, rare species are especially diverse in the Rocky Mountains, which support 81% of the known rare flora. And understanding rarity is critical for their conservation and their management, but for large-scale monitoring purposes, the distribution and diversity of common species is essential. So particularly for our province, where 58% is covered by boreal forest. And the majority of species occurring in this natural region are neither rare nor unique to this region. So for us, it's less about rarity and more about building a foundation for our biodiversity and then continuing to track changes in it. So second was they are speciose, and there are over 600 species of bryophytes in the province. This is of particular importance in ecosystems where other taxa are limited, such as the Canadian Shield. So here, the terrain is characterized by exposed bedrock and a landscape that's sort of left scarred by glaciers. Here, bryophytes and lichens can grow in areas where other plants can't, such as rock faces. And of course, bryophytes continue to dominate in black spruce bogs here as well. So this leads me to the next topic of gathering baseline data and why I think it's important. I am grateful to be a part of so many wonderful projects, but one of my favorite tasks within the ABMI is also one of the most basic, expanding our knowledge of what grows where. It amazes me that we continue to document previously unknown records. And I am proud to say that I am a part of a team discovering the range of bryophyte diversity within our province. These undisturbed and pristine habitats are a rarity in this world. And by documenting the flora and fauna of these habitats, it helps us better understand the consequences of disturbance and changes, because then we can compare those disturbance areas to these reference habitats. So speaking of disturbance, how much of our province is impacted by human land use? A recent report on human footprint by ABMI can be found on our website. As of 2018, total human footprint was 30.3% in the province. ABMI defines human footprint as the visible alteration of native ecosystems to residential, recreational, agricultural, or industrial landscapes. So let's just briefly take a closer look. In the Canadian Shield natural region, less than 1% of human footprint. For the Boreal region, which, like I said, is the largest in our province, the 2018 human footprint was 18.8%. And this contrasts to the Parkland natural region, where human footprint occupied 78.2%. I really do encourage you to go to the website and check out the full report. So the next, are bryophytes efficient to sample? So this is debatable for some, but there are some solid pluses. So they're small and they're really easy to collect. You literally just go out there, you grab them and you stick them in a brown paper bag and you let them dry and you're good to go. Also, lots of species tend to grow together, lots of different species. And it's not uncommon to find a dozen species all growing in a sample the size of the palm of my hand. So here we have sort of a typical moss sample. And just in this photo alone, without even dissecting it, you can see that there are like at least 7 species in this handful. We actually, I think my record is 16. So they are efficient to collect, but they are not necessarily the most efficient to identify, because for the vast majority of species, they cannot be identified in the field. They require dissection and microscopic examination. But we do have the expertise in-house in order to achieve efficiency and accuracy with our identifications. Research has shown us that bryophytes are highly sensitive to anthropogenic activities such as seismic lines and well pads, forestry, agriculture, pollution, roads, but also natural disturbances such as flood or other hydrology changes, fire, forest age and composition, and elevation. They are a fairly well-known group of plants, although liverworts to a lesser degree, but overall the taxonomy is manageable. Not to mention, we are so lucky in this province to have 2 of some of the finest bryologists, Dr. Catherine Lafarge and Dr. Richard Kainers, at the University of Alberta and the Royal Alberta Museum. 2 amazing people who are always willing to lend their expertise and their experience. And they are both curators of 2 of the herbaria that hold outstanding bryophyte collections with large representations from Alberta and beyond. ABMI actually helps contribute significantly to these collections, and we're just in the middle right now of preparing to deposit hundreds of fantastic specimens to the Royal Alberta Museum. There is also a growing body of literature from world-renowned experts aiding in the identification of mosses in the province. So finally is variability. So although this criteria is important when selecting across the entire suite of flora and fauna that ABMI monitors, it is also applicable just within the bryophyte group itself. So populations range from millimeters to centimeters, but can easily spread up to several meters for established colonies. And of course, bryophyte-dominated peatlands operate on this much larger scale of kilometers. They exhibit various life strategies, ranging from the short-lived colonist or ephemeral populations to long-lived perennial dominance in forest and peatlands. So now I will link sort of what I've talked about in general about bryophytes, their biology and ecology and being a good indicator, and connect it to sort of ABMI, what ABMI does. First on how we collect the samples, and then what we do with the data and the products that ABMI provides. So I will give a brief overview of our field and laboratory protocols, but I will be focusing on the bryophyte component. I should mention that these field crews do an awful lot more than just collect mosses. Details of our procedures are available to anyone who is interested on our website. So in general, it follows a fairly simple process. We train the technologists who will go out and collect the samples. They get out there and collect them. They ship them back to Edmonton, where they are dried, frozen, and packaged. And then we spend the next year or so identifying them. The data and the specimens undergo a rigorous quality assurance and quality control process. And then with the final stage being the development of the ABMI tools and products. So ABMI field technologists head out to a selection of randomly selected sites out of the 1,656 sites from our systematic grid across the province. Again, there's so much more to this than I have the time to get into, so I'm just going to emphasize a couple points about the bryophyte part. So first is the in-field training. This is done over several days in the field in different habitats that they will be visiting with our in-house team of experts. And I have personally had the pleasure of getting to know some of these amazing folks that go out there firsthand, and I know how good they are at their job. So second is how they sample. Since bryophytes can't be identified in the field for the most part, the techs are trained to collect from different microhabitats, logs, or stumps, soil, wet depressions, rocks, trees, and other vertical structures. So they also know when to collect from a log. It's not good enough just to grab a clump of moss. They have to look at the side of the log versus underneath the log or on top, because you have different species growing in these different microhabitats. Same thing for a tree. They look at the base of a tree, but then they also need to look upwards where a completely different group of mosses like to hang out. It is, however, also important that they look for some more obvious morphological differences so that we don't overcollect the same common species again and again. So for example, is the growth habit more like a tree or a staircase? Is it highly branched or very simple or not branched at all? Are there reproductive structures? Which shape are they? What color? Which way are they oriented? Are the spore capsules straight, curved, smoothed, or wrinkled? If you can see the teeth, the peristome teeth, what are the teeth doing? Now, this kind of morphological stuff is something I could talk about for ages. It's what I do for a living. I love it, but I have to move on. So once the samples come back to Edmonton, they are carefully dried and frozen. And then those same technicians who were out all summer come back to the lab, and they spend a month with us sorting and packaging the raw samples, removing any of the extraneous duplicates and identifying some of the common species. And then finally, my lovely teammate, Terry Hill, and I identified the samples with the utmost emphasis on generating an accurate data set. And here are some of the things that ABMI does with that data set. So the first one I want to talk about is our biodiversity browser. If you haven't checked this out, please do. It's fun. I use it all the time, not just for mosses, but all sorts of things. I saw a bird the other day. I looked it up and I was like, oh, does it occur here all the time? And yeah, it's great. So to illustrate the biodiversity browser, I'm going to compare two very different species. One is the fairly well-known Canada lynx, and the other is this little liverwort called the Heller's notchwort. So the lynx is a medium-sized cat. It's very cute, considered a snowshoe hare specialist, where the lynx abundance is directly related to hare abundance. Heller's notchwort is also super adorable, in my opinion. It's a really tiny plant, only about 1 millimeter tall, and it's found on really well-decaying logs. It produces these little specialized shoots, which you can see on the bottom left there, that stick up like little matchsticks, but only smaller. And they're topped with these reddish purple propagules that are called gemmy. So those little propagules, they break off of the parent plant, and then a clone of that parent plant can grow. Bryophytes do this all the time. It's very cool. So the first section in the biodiversity browser is habitat and human footprint. So when we look at these two, they both prefer forested habitats. They occur in the Rocky Mountains, foothills, Canadian Shield, and boreal forest natural regions. They also occur in almost all forest and vegetation types in the forested region and are virtually absent from all human footprint types. However, unlike the lynx, relative abundance is highest in older stands, particularly in white spruce and mixed wood. And up until a decade ago, once ABMI started their ecosystem health monitoring program, only a handful of records of Heller's notchwort from Alberta would have been documented. This map would have had literally like 2 or 3 dots on it. So the next section I'll talk about is impacts of human footprint. Here, ABMI presents predicted changes to relative abundance by sector. So the relative abundance of the Canada lynx is predicted to be higher in the energy and forestry footprints and lower in the urban, industrial, and agricultural footprints, and transportation to a lesser degree. Whereas the Heller's notchwort is predicted to be lower in all energy sectors. The last thing I'll talk about here are the predicted relative abundance maps. So here, I'm going to show the difference maps. The Canada lynx relative abundance is predicted to be lower in the pink parts and higher in the green parts. For Heller's notchwort, the relative abundance is predicted to be lower when compared to the reference conditions, basically throughout most of its range, except for a few little green spots. I'm not really sure what's going on there, but I'm looking forward to checking it out. So what does all of this tell us? Firstly, I hope the message here that you get is that you cannot just monitor one species to understand the complexities of our changing ecosystem, that different taxa respond differently, both across groups and within groups. Secondly, in this example, you can pinpoint these different sensitivities, where Heller's notchwort is highly sensitive and an indicator of all disturbance types, grows on these old logs and can be used as an indicator of old growth forests. So these individual species profiles are great and for species-specific responses, but ABMI was also designed to detect large changes in biodiversity across Alberta. So to do this, ABMI scientists created the Biodiversity Intactness Index to better understand how the suitability of biodiversity habitat has been modified. So it ranges from 0 to 100, where a high intactness score represents a current abundance of a species that is equal to what would be expected under their reference conditions. So this intactness score declines when a species abundance deviates, either positively or negatively, from its expected value. So I'm going to give you an example of this in use. So there is a forest management area in the northeast portion of the province that covers 10% of Alberta. It is a mix of harvestable and non-harvestable areas, as well as areas that are leased for oil and gas. The ABMI visited 154 permanent sites and collected data on 684 species across our 6 terrestrial groups, all the way from mammals down to mosses. And they also measured human footprint. So as of 2017, human footprint within this forest management area covered 8.5%, which was an increase of 4.8 since 1999. So what does this mean for the habitat suitability for the biodiversity of the region? Well, the ABMI Intactness Index ranged from 90.3% intact for birds and up to 96.6% intact for mammals. The overall average intactness score was 94.6%. But let's look back at our little friend, the Heller's notchwort, a species that we know is sensitive to human footprint. It had one of the biggest drops in habitat suitability, dropping nearly 2.5% in 5 years. So responsible forest management requires clear understanding of how forestry operations impact our biodiversity, and monitoring can help identify opportunities for improvement. And it's all about this data that is free from government or industry bias, that it's scientifically credible, where the results, tools, and products are made readily accessible, and the methods that we use are transparent. These are all the principles that ABMI is founded on. I love the idea that there's this single bryophyte collection and identification that ends up contributing to provincial biodiversity maps and helps predict changes to the health of our ecosystems. Watching that sample go through the ABMI life cycle, coming out the other end as this intactness index. But of course, it's not just moss or the moose or the warbler or the mite. It is the suite of taxa together that is ABMI's strength. So I am out of time for today, but please feel free to contact me if you have any questions. Of course, please poke around on the website, abmi.ca, and hopefully the next time you are out for a walk and you notice that small little green patch of moss growing between the cracks, you might just think about what information is hidden in that moss if we just got to know it a little bit better, because it is in fact our nature to know. Thank you.